All right, so here we're going to talk about the metabolism of fructose and galactose. These are the other two monosaccharides that your body can absorb from your diet, sugars. So in general, you have three monosaccharides that your body can absorb from your food. You have glucose, fructose, and galactose. And what you can see hopefully from this simple chart is that glucose is going to become glucose 6-phosphate, and it has the ability to go through glycolysis and become pyruvate, or it can go to storage and become glycogen. Now this is different than fructose. Fructose only has one fate, and that's to go through glycolysis. So if you take in fructose, you're going to have to push through glycolysis and make energy. And then your other sugars that you take in will go to storage. Galactose also can go to storage, or it can feed back into the glucose pathway to go through glycolysis. Now, fructose is going to come from different sources. You can have sucrose, which can be broken down into glucose and fructose. You can have dietary sources of fructose directly from honey, or you can make your own fructose through something called the polyol pathway. All three of these are going to feed into glycolysis. Fructose metabolism is going to, in the muscle, we have a hexokinase that can convert fructose to fructose 6-phosphate, but this is very minor. Your muscles do not like fructose. Fructokinase C is the version in the liver, and this is going to be the major site of fructose metabolism. So your fructokinase, similar to hexokinase, added a phosphate to your glucose during glycolysis. Fructokinase C is going to add a phosphate to fructose, and this is on carbon position 1 here, and you're going to get fructose 1-phosphate. And then fructose 1-phosphate has to be metabolized. If you have accumulation of fructose 1-phosphate, this can lead to diseases. So basically, the next step after you have fructose 1-phosphate is that you're going to have aldolase work on fructose. And it's important to pause here to talk about the different forms of aldolase. Aldolase A is going to be in most cells. Aldolase C is going to be in the brain. Now remember glycolysis. In glycolysis you're going to form fructose 1,6-bisphosphate. And aldolase A and aldolase C are going to be able to make those into the triose phosphate. And that's the glyceraldehyde 3-phosphate and the DHAP. Now, aldolase B is expressed mainly in the liver, and this can hydrolyze fructose 1-phosphate. So remember, the liver is going to have aldolase A and aldolase B. Aldolase B can also perform hydrolysis and glycolysis as well, but it is going to be very important for metabolism of fructose. Okay, so let's talk a little bit about what's going on here. We have glucose that we talked about. It's going to get phosphorylated by hexokinase, become glucose 1-phosphate. This is then going to go through glycolysis, and eventually you can go to the TCA cycle to make ATP. Fructose is also going to enter the liver, and it's going to be worked on by fructokinase. It's going to be phosphorylated at that 1 position. You're going to require aldolase B to generate glyceraldehyde and dihydroxyacetone phosphate. Now, dihydroxyacetone phosphate feeds right into glycolysis and goes to the TCA cycle. Your glyceraldehyde has to add a phosphate to it through a triose kinase, and you get glyceraldehyde 3-phosphate which also can feed into glycolysis. Now, fructose, the consumption of fructose, high fructose corn syrup, has been correlated with the development of diabetes, obesity, and metabolic syndrome. And metabolism of fructose is going to lead to an increase in serum uric acid. Now, fructokinase is going to lead the use of fructokinase is going to deplete your ATP stores. Because it's using the ATP, of course, to phosphorylate fructose. And fructokinase has no feedback inhibition. So as much fructose as you eat, fructokinase will add a phosphate and you'll metabolize those carbons in fructose. Now the hepatocyte is going to be the primary cell that metabolizes fructose. So when you have a decrease in ATP, this is going to lead to an increase in AMP, and this leads to activation of your purine nucleotide catabolic enzyme called AMP deaminase. Now, AMP deaminase is going to degrade AMP into uric acid. Oh, I should do this, sorry. Now, uric acid gets elevated in the blood during both hypertension and obesity, and gout is also associated with increases of uric acid. So again, it's important to understand that fructose, when it is part of your diet, it has to be metabolized. It's sort of almost a feed forward because your fructokinase is not regulated. And so it's going to keep making fructose 1-phosphate. And it's going to keep adding those phosphates to fructose, and that's going to increase AMP, and that's going to activate the destruction of AMP into uric acid. Now, there are some fructose metabolism disorders. We have essential fructosuria, and this is going to be caused by a lack of fructokinase in the liver, in the pancreas, as well as in kidney cortex. This is benign. Basically, if you have essential fructosuria, you're just going to simply excrete fructose. You're not going to metabolize it. Now, the more important one is hereditary fructose intolerance, and this is where you lack aldolase B. Now, aldolase B is going to have to metabolize fructose 1-phosphate. And if you can't do that, then you're going to accumulate fructose 1-phosphate, and there's nothing else your cell can do to this. So fructose 1-phosphate is going to accumulate, and it's going to lead to severe hypoglycemia and vomiting, and it can be lethal because it gets trapped in fructose 1-phosphate. And anytime something gets trapped, that's not a good thing because your body doesn't do well with accumulation of things. And so you must be on a diet that is lacking fructose. There is also fructose 1,6 bisphosphate deficiency, and this is hereditary autosomal recessive. And this is going to be a loss of function, it's going to be an impaired gluconeogenesis, and you're going to have episodes of hypoglycemia because of a lack of your liver to be able to make glucose. This can also be lethal in infants. Now, one other thing to keep in mind with glucose is you can actually make, or fructose, you can actually make it. This occurs when you have too much glucose. So again, going back to diet, if you take in too much glucose, there's only so many places your glucose can go. We talked about you're going to take up glucose, and you're going to get glucose 6-phosphate, and this can go to glycolysis, and this can go to glycogen, and this can go to fatty acids, and this can also go to making fructose. And so you can imagine, why does your cell want to make fructose? Well, it doesn't. It just means there's too much glucose. And so when it's going to make fructose, it's going to eventually have to push this glucose or fructose back into glycolysis. So it's almost like it's a temporary way to push those carbons in one direction, but you're ultimately going to have to go back to glycolysis. However, let's just look at this. What's happening here is you have your glucose, and then you're going to have an aldolase reductase, in which you're going to take NADPH and reduce it to NADP, and this is going to cause sorbitol production, polyol. This is why this is called the polyol pathway. And then from sorbitol, you're going to have a sorbitol dehydrogenase. Yes, and that's going to take NAD and make NADH. And so you're still going to be accumulating in the cytosol NADH regardless if you go down this road or down glycolysis. The side note is this can lead to deposits in the eye. And so it's all a big giant circle. If you don't have enough NADH, so if you have a high NADH to NAD ratio, this means you have low NAD. And so you need NAD in order to convert sorbitol to fructose. If you lack NAD, you're going to accumulate sorbitol, and this can lead to these sorbitol being deposited in eyes, and this is something that's seen in obese and type 2 diabetic patients. So again, it just goes back to the availability of these intermediates to move around electrons, to move your carbons through different cycles in order to prevent accumulation of these carbon sources and potentially disease. All right, so let's talk about galactose now. Galactose is going to be picked up by your liver primarily, and it has a galactokinase, like a fructokinase, and a glucokinase. And again, you're going to add to that one phosphate, that one carbon phosphate. Then you have galactose 1-phosphate, and galactose 1-phosphate is going to need a UDP glucose, and you're basically going to take the galactose phosphate and transfer it from galactose to UDP glucose to generate glucose 1-phosphate. And this is a galactose 1-phosphate urididyl transferase. And then once you have glucose 1-phosphate, you can either make glucose 6-phosphate. You can also go on to glycolysis. You can make glucose. You can make glycogen stores, whatever you want to do with your glucose. Now your UDP galactose is going to be recycled into UDP glucose, and UDP glucose can then pick up another phosphate, or UDP glucose has other fates. So unlike our fructose, the potential for other things to happen for galactose is there. Galactose can become UDP glucose. UDP glucose can get through to glycolysis, or in the liver can be used for gluconeogenesis, or you can go on to glycogen stores. So for galactose, you don't have to metabolize it through all the way through the glycolysis. There are other mechanisms. But galactose also does have some disorders. So we have classic galactosemia, which is type 1, and this is a profound deficiency. This is going to be a galactose 1-phosphate urididyl transfer deficiency. And so again, you get stuck in this galactose 1-phosphate. This is autosomal recessive and presents always in neonatal periods. You're going to have failure to thrive, bleeding problems, potential sepsis, and you have to eliminate lactose from the diet because remember, lactose is going to be broken down into glucose plus galactose. Type 2 galactosemia is going to be a deficiency in that first step, galactokinase. And so galactose is a little bit different than fructose. If you had an accumulation of fructose, you just excrete it. For galactose, it's different. You're going to accumulate galactose in the blood and in the tissues, and infants can present with cataracts. When you remove the galactose from the diet, the cataracts will also be removed. This is not a lethal genetic deficiency like the urididyl transferase, but it's still you have to remove galactose from the diet. And then there's a type 3 galactosemia, and this is a deficiency of that galactose 4 epimerase. There are two different isoforms of this. One is going to affect red and white blood cells, and this is going to be very rare. There are actually only 8 known cases. You get multiple tissues affected, and it's similar to our classic galactosemia. Glucose, UDP glucose is important, and getting stuck at UDP galactose, again, you're just accumulating UDP galactose, and your cells have can't do anything with that.